Hello, everyone. Thanks for joining us today. We will wait another minute or two to allow everyone to join, and now we will get started. And also, while waiting, you may want to participate in the posted polls. The results of these polls will be available to all of us at the end of the presentation. Also, please do ask your questions throughout the talk by typing them into the chat box. We will address them at the end of the talk as well. We will give it another 30 seconds and then we will start. So, welcome everyone again to today's webinar titled A Novel High-Resolution Perfusion Platform for Studying Insulin Release Dynamics of Single Human Pancreatic Islets. My name is Burcak Yesilag. I lead islet biology activities at InSphero. I'm located in Zürich. In this talk, we will present our collaboration with ETH Zürich. That's why I'm joined here by Patrick Misun, the postdoc who led the projects at the ETH Zürich.
Thanks, Burcak, for the introduction, and welcome, everyone.
Perfect. So today we will be talking about multiple different topics. So we will start our talk by explaining the limitations of in vitro translation of dynamic insulin secretion assays. We will then talk about how we generated our human islet microtissue model that we use in these studies. We will move on to Dan Patrick's part, where he will explain hanging drop-based microfluidic perifusion platform that they engineered in the bioengineering laboratory of ETH Zurich. Then he will present the work he has done by perfusing single islet microtissues. We will then close with a summary and an outlook and ongoing activities on our site. So let's get started. I would like to give a brief recap of the pancreas and the insulin-secreting pancreatic beta cells, mainly for the engineers among us. And I'll try to be very brief with this. So the pancreas itself, it is a dual function organ, and it encompasses both endocrine and exocrine functions. The exocrine pancreas makes up to around 98% of the pancreatic mass, and it's in adult humans, and it's responsible for secretion of digestive enzymes into the intestine. The endocrine pancreas, on the other hand, it takes much less space. It's around 1 to 2% of the total pancreatic mass. And these endocrine cells, they take the form of small clusters that are distributed throughout the organ. These clusters are called the islets of Langerhans. So they can be actually also observed phenotypically distinct from the rest of the pancreas. And they are composed of multiple different endocrine cell types, including, or mostly abundant, the insulin-producing beta cells, the glucagon-producing alpha cells, and the somatostatin-producing delta cells. But there exist other endocrine cell types in the pancreas as well. The loss of insulin-producing beta cells, or insufficient secretion of insulin from these cells, leads to the most common metabolic disease of mankind, diabetes. And the beta cells, they secrete insulin not in a passive way. It's a very active, very well-regulated event, and it is biphasic and oscillatory. So in response to a glucose stimulation, the beta cells respond by releasing the readily releasable pool, which results in the first phase insulin secretion. Then they start to release from their reserve pools, that is resulting in the second phase of insulin secretion. In impaired glucose tolerance or type 2 diabetes, the insulin secretion process is highly impaired by blunted first phase and oscillations of the second phase of insulin secretion. These studies, the dynamic insulin secretion, can be monitored or studied in vitro by perifusion systems. And the islets themselves, the native islets, presents multiple challenges to these studies. So I would like to first talk about what these limitations are. So the native islets, they can be easily isolated, or maybe not much easily, but quite efficiently isolated from the pancreas in the forms of pancreatic islet clusters. And they do present some inherent size variation. This can vary from a couple of micrometers in size to 300, 400 micrometers in size in adult individuals, and adult healthy individuals actually. And they also can present very heterogeneous cellular composition. Heterogeneity in islet composition is generally more apparent in higher mammals such as humans. Also, they can present variable purity. This is obviously not an inherent property of an islet, but it happens after isolation. And it could be that some islets are more pure than others, and some islets may have endocrine fractions that are inside the islets themselves. And due to the bigger size and probably also the exocrine contamination, the islets, they can display a rapid decline in functionality and viability during ex vivo culture. So typically, the human islets needs to be used fresh, or their functionality will decline in a matter of 2 to 3 days. Because of all these limitations I've just mentioned, generally the perifusion of islet pools may be required to tackle this inherent variability, but also the perifusion of islet pools may be required to achieve quantifiable insulin concentrations by the assay system that is used. However, the pools results in decreased resolution due to uncoordinated secretion of different hormones from the islets. And this is because there is no factors that synchronize these islets ex vivo, whereas multiple factors from liver, intestine, and the brain does contribute to the synchronization of the islets in vivo. Additionally, I would like to mention a lot of the perifusion studies in the past has been done by rodent islets. Rodents obviously present a very good model system to work on islet biology. However, as mentioned by Rorsman and Ashcroft in their review, despite human and rodent beta cells sharing many features, it should not be taken that they are identical. There are multiple differences between the two species. To name a few, we do have very different feeding times, feeding behaviors, and diet. Also, inside the beta cell, there is a different complement of ion channels, receptors, transporters, and exocytotic proteins between these two species. So ideally, for studying islet biology and perifusion in vitro setting, we would want a model system made from human cells. We would want these islets to be homogeneous in size, composition, and function. We would want them to be stable in long-term cell culture in terms of functionality and viability. Of course, we also, they should respond in an established way to pharmaceutical manipulation, and they should display physiologically relevant dynamic insulin secretion to be used successfully in single islet perifusion systems in vitro or ex vivo. So I would like to mention quickly how we addressed on making this, to make this ideal islet model. So we do take isolated human islets that are coming from human pancreas. We always work with very pure preparations, generally above 85 to 90% purity line. Then we very gently, enzymatically, and basically mechanically process them into single cell suspension. Then we reaggregate them into homogeneous cell clusters that we call human islet microtissues. We do this by using our hanging drop multi-plate culture plates, multi-well culture plates. They encompass 96 wells of hanging drops, and basically we seed the cell suspension into each of these hanging drops, and the cells, they spontaneously aggregate into these clusters in around 4 to 5 days. Then we transfer these newly forming islets into the receiver assay plate, which we call Accura 96, and culture them there for about a month, which they sustain viability and functionality. The final product would be a 96 valve plate with 1 islet per valve, and the islet size that we aim for is around 150 micrometer. And we generally make sure to control this size very well between different lots of production, aiming for nothing smaller than 135 micrometer and nothing bigger than 181 micrometer in diameter. When we go through this reaggregation process, we have discovered that we have around 55% beta cells, around 35% alpha cells, a bit less than 10% somatostatin producing delta cells, and 2 to 3% of other cells, which may be epsilon cells or pancreatic polypeptide cells or any other cells such as immune cells or contamination that is left in these tissues. This percentages really well represent what has been previously published and what is expected from a human, average human islet population. So the native-like cellular composition is preserved. We also saw that the tissue architecture is distinct when we do rodent and human islet market tissues, and this corresponds to the differences between rodent and human islets. You can see that in the red islets, this is a pancreatic section, there is a arrangement that is a beta cell with a beta cell core and a non-beta cell mantle, whereas in humans this is different. It is generally a trilaminar structure that is beta cells in between and non-beta cells generally pushed to the outside that is folded on top of itself. And when we make our microtissues, we do see the similar structure. So for the red islet microtissues, we have a beta cell core and a non-beta cell mantle, just like the red pancreatic islets. And then in the human islet microtissues, we do see a more intermingled distribution with generally alpha cells and beta cells arranging themselves in distinct groups. Another problem that the islet biologists face generally is the variation in islet function or beta cell function between different isolations. And we know that this is generally due to the quality of the islet prep. And once we go through the reaggregation process, and we know there happens multiple differences after the reaggregation, such as exclusion of the contaminating exocrine cells, or probably reformation of this islet architecture, we actually obtain a very robust glucose-stimulated insulin secretion from all the donors we have tested. So far, we have tested 143 donors in our laboratory. I show here 10 different donors that are consecutively tested of these 143. And what we see when we do these experiments, that we see very little variation in stimulated insulin secretion at 16.7 millimolar glucose. This is around 2.5-fold between different donors we tested. We see some intermediate variation in the basal insulin secretion. This can reach up to 4-fold between different donors we tested. And we see more variation at intermediate glucose concentrations, such as 5.5 or 11 millimolar glucose. And this may be due to different affinities of rate-limiting enzymes, such as glucokinase and glucose transporters on the cell surface of different donors, and a well-known personal variation. And of course, all the donors we have tested are healthy, or is not diabetic. So if we were testing donors in insulin resistance or diabetic, we expect obviously more difference in these preparations. Our islets, after reaggregation, also they do sustain a functionality very long, around 28 days that we tested in long-term cell culture. The islet size and the ATP content stays very stable throughout this time. However, the insulin content, we observed that it increases by culture time. It may be partially the recovery of lost insulin during aggregation and dispersion, but it may be also the lack of stimulation throughout this culture period. However, this doesn't influence the glucose-stimulated insulin secretion, and we see a very stable secretion for the culture period, and each donor seems to have a very distinct phenotype of secretion, which stays stable again throughout this time. This variation here, when we combine 6 individual donors, is of course more high, but as you can see here, individually inside each prep, the variation is quite minimal because of the uniformity of the islets. We also established that our islets, they respond to different pharmaceutical stimuli in an expected way. Here we showed 2 different stimulation with Insulin Secretogogs. One is exandin-4, a GLP-1 receptor agonist, and the other is tolbutamide, a sulfonylurea class of compound. For GLP-1 receptor agonists, such as exandin-4, we expect the response to be glucose dependent. These compounds or these hormones, they tend to they work by augmenting glucose dependent pathways in the cells, and therefore only work better in increasing glucose concentrations. However, sulfonylureas, they do a bit more of an electrophysiological stimulation by closure of potassium channels on the cell surface, and therefore their action is more independent of glucose, and they do actually work better in intermediate glucose concentrations in our hands and less in higher glucose concentrations, probably because glucose then dominates the overall secretion capacity. So I showed you now that we have displayed that this model system made from human cells is homogeneous in size, composition, and function, and it is stable in long-term culture. And we were able to establish responses to pharmaceutical manipulation. However, at this point, we did not know whether these islets do also secrete physiologically relevant insulin, dynamic insulin, or display physiologically relevant dynamic insulin secretion. Therefore, we have collaborated with ETH Zürich to analyze these properties. So I'm passing the presentation to Patrick now.
Thanks very much, Burcak. In the next couple of slides, I would like to present to you a hanging drop-based eyelet perfusion system, which we developed at the bioengineering laboratory at ETH Zurich, which allows for studying the dynamics of insulin release at a single eyelet level and at high temporal resolution. We can not only use such an eyelet perfusion system to assess eyelet functionality, but also to study effects of antidiabetic compounds. And as we learned from Burcak's presentation, we have now access to a standardized human eyelet model that we can stimulate in glucose-stimulated insulin secretion assays. And you can see here a nice dose-response curve. But what we don't see is how dynamically islets are secreting insulin, which appears in 2 very distinct phases. Now, in order to resolve this dynamic insulin release, we need dedicated perfusion systems which allow for a continuous medium sampling from the islets. Now, what are eyelet perfusion systems and what are their design requirements? Eyelet perfusion systems are very simple fluidic devices which have inlet ports, a perfusion chamber which hosts the eyelet, and an outlet port. Now I'm going to list a couple of design requirements that we think are important when designing eyelet perfusion systems. The first one is to design a miniaturized perfusion chamber which allows for a precise but also stable positioning of a single eyelet microtissue. Also, we have to make sure that we allow for an easy access to the perfusion chamber for easy loading of the islet microtissue, but also easy harvesting of the tissue for endpoint analysis. The second point is to make sure that we have a precise fluidic control, but also an optimal flow profile to achieve short delays and high spatiotemporal resolution. So in this example, for example, the eyelet is sort of shielded from the active perfusion flow, which is subideal. And what I mean by having an optimal flow profile is something I'm going to show you in the next couple of slides. The third point is to allow for an immediate and also undiluted transport of the secreted analytes to also be able to detect very small quantities of insulin that was released from a single islet. The fourth point is to achieve liquid sampling at high frequencies in order to achieve high temporal resolution sampling. Also, we have to make sure that we keep the system as simple as possible. So on the setup side, as well as on the readout side for the analysis, to make the system accessible for researchers across different laboratories. Now we tried to address all of these requirements, and we came up with our GSS Flow CSS Platform, which is based on a microfluidic hanging drop chip, where we connect a single hanging drop to an inflow port, and we have a sampling port from which we can sample the media that can then be analyzed. And with this setup, we are able to resolve the insulin release footprint of a single islet. Now let's have a closer look at the layout of our chip. The chip is based on a surface pattern PDMS substrate with a small rim structure, which acts as a fluid phase guide. You see the liquid represented in blue, and we have an inlet port on the left-hand side. We have a round circular hanging drop compartment in the middle of the chip. You also see the location of the eyelet microtissue, and we have an outlet port. If we now have a look at the cross-section, we see again the rim structure, which acts as a capillary and hydrophobic stop. You see the position of the eyelet microtissue at the bottom tip of the drop, which we keep in a very small culture volume of roughly 6.5 microliters. So this is a completely open microfluidic chip design, which allows for an easy loading, but also harvesting of the eyelet microtissues. And the shape of the hanging drops make sure that we have a precise location of an individual eyelet located at the bottom tip of such a drop. Now this is the experimental setup, and you can see we can now connect different syringe pumps that are preloaded with different glucose-enriched buffer solutions, but also compounds, for example. And we perfuse at a constant rate at 15 microliters per minute, and we actively withdraw the media with the peristaltic pump with the same flow rate. And the media is then sampled via a robotic arm into an assay well plate that can then be directly used for ELISA quantification. So we're using here a simple standard ELISA. We also have a microscope to track the position of the eyelet microtissue, and we then implemented a feedback algorithm that fine adjusts the syringe inflow rates to make sure that we can maintain the drop shape and volume. Now, what is the advantage of having an open microfluidic chip system versus a closed one? So on the left-hand side, you can see an open microfluidic chip architecture, such as the hanging drop chip. We have here a liquid air interface, which gives us a slip boundary condition at the liquid air interface, meaning that the molecules are dragged with the flow from this liquid air interface directly to the outlet port in a rather rapid fashion. In the closed microfluidic systems, however, we have a substrate liquid interface, which gives us a non-slip boundary condition, meaning that, for example, if insulin was released from the eyelet, these molecules have no flow and they can diffuse into the bulk and then can then be dragged with the flow towards the outlet with a certain delay. What does this give us? So in the open microfluidic chip system, with the slip boundary, we have now a rapid liquid turnover we have also less dilutions and a minimal dispersion of the analytes that are secreted from a pancreatic islet. In the closed microfluidic scenario with a no slip boundary condition that I show on the right-hand side, we have a slower liquid turnover, we have more dilutions, and we have a greater dispersion of the analytes that are secreted from an islet microtissue. Here I would like to show you how fast we can switch between one and the other buffer solution. Here we switch from DI water to a fluorescent dye. You see the red curve, this is the measurement, and the dashed line is our simulation data. And you can see that to replace the media in our hanging drop device, we need roughly 2.5 to 3 minutes for a complete washout. Now, however, when we look at what happens around the eyelet, which is located at the liquid air interface, which is the solid line, we see that the media switches already happens within 13 seconds. So we have a very fast and rapid liquid turnover around the eyelets. The eyelets also experience only minimal shear stress in our hanging drops. And since the eyelet is located at the liquid air interface, we also could achieve an optimal oxygenation of the media as well as of the eyelet, which improves not only eyelet viability, but also functionality of the eyelets. Now coming back to the precise fluid control and the optimal flow profile. What we did here is we simulated the flow path of two different scenarios in our chip, having a slip boundary condition represented in black, and a non-slip boundary condition represented in blue, which is the closed microfluidic chip scenario. And then we simulated an insulin release burst from an eyelet microtissue that is located at the bottom tip of this hanging drop structure. And we are measuring the concentration that we get on the outlet of this device. You can see here the respective well plate positions. So we continuously perfuse at the flow rate of 15 microliters per second. And we sample into every single well for 30 seconds. So we collect 7.5 microliters in every well. And we also here measure the average concentration of insulin that we get. Now I'm going to go through the sequence. So we start at 30 seconds. And you can see now that already after 30 seconds, most of the insulin that was released in the open microfluidic chip environment was dragged and passed the outlet, whereas in the no slip boundary condition, so in a closed microfluidic system represented in blue, there is still a lot of insulin present in the hanging drop compartments. And you see that the washout takes way longer. So what do we learn from this? In an open microfluidic chip system, such as the microfluidic hanging drops, we have less dilution, we have a reduced dispersion, and with this we have an increased spatial temporal resolution, which is important to record or resolve very fast occurring events. So now we have this setup which enables us to resolve the insulin release footprint of a single eyelet microtissue. You can see at the bottom the stimulation protocol. So we switch from low glucose to high glucose and then back to low glucose. And we can now subdivide the curve into different phases. So we have a baseline, then we enter a sharp dip right after the stimulation. Then we enter the first phase of insulin release. Then we see a pulsatile sustained second phase, and we see a post-stimulation phase. We can now extract so many parameters from a single curve. We can also look at the timings. We can, for example, see how long does it take to reach the peak secretion? What is the duration of the first phase? What is the frequency of the oscillations? And what is the relaxation time, so the time it takes for an eyelet to relax from the glucose stimulus? As I mentioned, we can now extract these parameters and we can look at the average insulin secretion and subdivide this and look at it in every phase represented in red. So this is data that we took directly from the curve that you see on the left-hand side. And in gray, we can also have a look and see what is the total amount of insulin that was released in the different phases, which is sort of the integral of the curve that you see on the left-hand side, subdivided into the different phases. But we can also have a look at the timings. And what we observed is that some of the timing parameters highly depend on the donor, whereas others are highly preserved across different donors. So for example, the peak secretion highly varies on the donor, whereas the T response, the time to reach the peak, or the duration of the first phase is highly preserved across different donors. Also, the T relaxation time, for example, again, depends more on the donor of the islets. We can now also have a look and see how compounds modulate this insulin release footprint. Here, for example, exendin 4, which only stimulates insulin release in the second phase, but does not change the first phase of insulin release. Whereas tolbutamide, for example, already elevates insulin release in the pre-stimulation phase, then we see again a well-pronounced dip, and we see that the first secretion phase is reduced. We think that this is due to the fact that the partially ready releasable pool of insulin already got depleted in the pre-stimulation phase at low glucose concentrations. And this is the effect that we see then in the first phase. We also see that in the second phase, tolbutamide didn't change the stimulation pattern too much. And we see again an elevated insulin release at the post-stimulation phase at low glucose concentrations. And also here we can now extract these compound-regulated parameters and dissect them into the different phases. And what we see is, again, that exendin predominantly elevated insulin release in the second phase and didn't change it in the first phase or in the peak secretion. And we see a small reduction in the post-stimulation phase. Now, with tolbutamide, we see an elevated insulin release in the pre-stimulation phase. As a consequence, we see a reduced peak secretion and also a reduction in the first phase of insulin release. We see that the second phase is not changed, and we see that in the post-stimulation phase, again, we have an increase of insulin release. Here, as one of my last slides, I would like to compare and show you single versus pooled islet perfusion. We observed multiple times that when we pool islets, we see a reduced first phase and also second phase insulin release when we calculate the insulin secretion per islet equivalence. And we also see this when we stimulate with compounds such as exendin 4 in that case. On the right-hand side, you can again see that the first phase is reduced and also partially the second phase is reduced. But what you also see is that we don't get any information about insulin oscillations when in the pooled islet scenario. This was most likely a result of the averaging of asynchronous insulin secretion patterns of individual islets. With this, I'm already at the end of my talk, and I would like to hand back over to Burcak.
I'll just summarize our platform and the results before closing and going to our outlook. As you have heard, we have engineered a standardized primary islet model that is uniform in size, cellular composition, and function. It displays native-like cellular architecture and cellular composition. They are functionally robust, and they retain this functional robustness for almost a month in cell culture. So this model allowed us to do now single islet perifusion studies because of its uniformity and reproducibility between different islets within a single production. And the bioengineering laboratory in ETH Zurich then engineered a perfusion system that is based on a hanging drop chip that provides rapid medium switching, therefore results in minimal analyte dispersion, and which can sustain short sampling intervals. And therefore, we can also do precise quantification of insulin secretion from these islet marker tissues. And it's compatible with standard measurements such as sensitive insulin ELISAs that are already in the market. By combining these two models, we were able to have a platform that allows of perfusion of single primary human islets at high temporal resolution. And we were able to see a reproducible biphasic and oscillatory insulin secretion profile among different islets and among different donors. And again, to mention, this type of temporal resolution, we believe, is only feasible when one perfuses single islets, because in islet pools, one would have uncoordinated functioning, therefore blunting of these responses. To name a few of the biological findings we have observed is that we saw that the first phase insulin secretion was increased around 35-fold over the basal, with peak increase being almost 64-fold over the basal insulin secretion. And during the second phase, we saw sharp oscillations in most donors, and this indicates intact cellular communication, because oscillations would require every beta cell to be communicating with each other within a single islet. Then we also saw higher secretion rates from islets under perfusion compared to islets in static conditions, but both basal and stimulated insulin secretion increased in perfusion, therefore resulting in similar fold induction. We believe this may be related to the lack of accumulation of inhibitory factors such as somatostatin. Also, we have seen some parameters were preserved among different donors, whereas the others were more variable. For preserved parameters, we have seen initial time to respond to glucose, the time to reach to peak secretion, and the duration of the first phase was very preserved. And the average secretion rate, the peak secretion rate, and the relaxation time, as well as the frequency and the intensity of oscillations, was quite variable among different donors we have looked at. Having set up the two model systems and created this platform, we now would like to enter disease modeling and drug development. And for this, we would like to first look at how dynamic insulin secretion is influenced by stress, using different injuries such as cytokine-induced islet stress, lymphocyte-induced islet stress, or other metabolic injuries such as glucotoxicity, glucolipotoxicity, and lipotoxicity. And we would like to then try recovery of these impaired dynamic insulin secretion by using acute and chronic pharmaceutical intervention. Independent of these topics, we are also very much interested in assessment of cell maturity in stem cell-derived beta cells. And also at the ETH Zürich site, there's ongoing studies to increase the output of the system by making the next version of this platform, which includes 4 perfusion chambers that can sustain perfusion of 4 different islet marker tissues simultaneously. So more details are presented in our paper that was recently published in February in Advanced BioSystems. So for people who are interested to see more data, I suggest to check out this publication. And by this, we came to the end of our talk, and I would like to thank everyone involved in these studies, and to, again, Patrick and the bioengineering laboratory, and Andreas Hirlemann, who is leading this laboratory. Thank you very much for your time. We can take questions now.